Please appreciate this presentation has been prepared with all sincerity and to the best of knowledge of students, teachers, and volunteers. We welcome words of encouragement and also suggestions for improvement. For use in academics or any kind of exams, please verify and correlate with your course books. For becoming an associate and/or sending contributions in different subjects, including storytelling, please get in touch with your brief profile at baskenjoy@gmail.com. So welcome back everyone. Today will be the last lecture for the fundamental unit of life topic for class 9 biology. In today's lecture, we'll be making the plant cell and animal cell. We'll be drawing their diagrams. We will be comparing and contrasting them, and we'll also take a look at cell division. So first up, we'll take a look at the animal cell. So these are the components of animal cell. So if by any chance you're not familiar with any of these cell organelles, I highly recommend watch the previous lecture, lesson number 3, to get to know about each of their functions. So first of all, we have the nucleus, then we have the plasma membrane, the cytoplasm, and then vacuoles. It's an S. We have multiple vacuoles in an animal cell. We have mitochondria, we have lysosomes, we have endoplasmic reticulum, and this was divided into smooth and rough, if you remember from the last lecture. And then we also have the Golgi apparatus. So now we'll quickly draw an animal cell. So an animal cell is basically a circle, an oval, whatever you want to call it. So then this outside layer is the plasma membrane. And then in the center, we will have the nucleus. In the center of the animal cell, we have the nucleus. And this is surrounded by the nuclear membrane. Okay, so the outer covering is the nuclear membrane. The inside is the nucleolus, and these two combined make the nucleus. Okay, and then around the nucleus, as you know, we have the endoplasmic reticulum. This is just a brief drawing of it. It doesn't have to be super fancy, but you can just make it a circle like this, just going around the nucleus, and this is the endoplasmic reticulum. So now we have two endoplasmic reticulums, as you must remember. This we will call the rough. The rough has ribosomes on it, and this is the smooth. So we have the rough ER, and this we have as the smooth ER. And these combined make the endoplasmic reticulum as a whole. And then on the rough ER, we have ribosomes, which synthesize the protein. And then we also have multiple vacuoles, but they're small in size. So this is basically one of the vacuoles. Well, you can make it plural. We'll just mark all of them vacuoles. These are the vacuoles. And then we have the mitochondria. We need that. It's the one which is going to produce the energy, and it has this wavy inner membrane. So we have the mitochondria. And then the last two things we need are the Golgi apparatus. This is also it's also sometimes called the Golgi body, or you can also simply say apparatus. They're the same thing. Either you say Golgi body or Golgi apparatus. And then lastly, we have our lysosomes, the cleanup crew of the cell. We have lysosomes. So this is the brief diagram of an animal cell. There are also some other minute cell organelles, but then these are the main cell organelles which any cell has. So we have the nucleus consisting of nuclear membrane and nucleolus. We have the plasma membrane covering the cell. We have the rough ER and smooth ER making the endoplasmic reticulum. We have multiple vacuoles. We have like 1, 2, 3. We have more than one vacuole, but they're small in size. They're really small in size. We have mitochondria. Sometimes they can also be 2 mitochondria or 3, so we can also make another one if needed. There can be multiple mitochondria. There's one Golgi body or Golgi apparatus, and there are lysosomes inside an animal cell. So now we have the animal cell out of the way. We will take a look at the plant cell. Now the components of the plant cell are the nucleus, plasma membrane, and you also should remember it also has a cell wall. The cell wall is outside the plasma membrane. It covers the plasma membrane. We have the cytoplasm. We have a single vacuole. It's an E, it's singular. So we only have one big vacuole. It will take up 50 to 90% of the space inside the cell. We have mitochondria, and actually plant cells do not have lysosomes. So you might ask, don't they need lysosomes to basically clean the cell up? But actually they don't require lysosomes, as first of all the cell wall already prevents material from entering the cell. So like I said earlier that in an animal cell, if there's a bacteria or something, the lysosomes will basically digest it, but the cell wall already prevents most of the things getting inside the cell, the big things which might do harm. So that's why they don't require lysosomes. But in case the vacuole might act as a lysosome, as the vacuole is so big, there's sometimes sections inside the vacuole which are dedicated to waste products. So sometimes when necessary, the vacuole of a plant cell might act as a lysosome, but in most cases plant cells don't actually have lysosomes. Well, they need the endoplasmic reticulum, the Golgi apparatus, and they also have plastids, or simply chloroplasts. So now we'll draw a diagram of the plant cell. So as the animal cell was like circular, the plant cell is more rectangular. It's more edgy due to the cell wall. The cell wall makes it rigid. So now we have the thick outer covering, the cell wall. Okay, this outer covering is the cell wall. This inner covering is the plasma membrane. And then the vacuole is so big in an animal cell that it pushes the nucleus out of the center. The vacuole becomes central and it pushes the nucleus, so the nucleus is off to the side inside an animal cell. So then this is the nucleus. I didn't label this is the nucleolus, this is the nuclear membrane. Okay, and then now we can draw the similar endoplasmic reticulum around here, around the nucleus. And then this one we'll call it rough ER. It has the ribosomes. So this one can be rough ER. This one can be smooth endoplasmic reticulum. And then we have plastids or chloroplasts. So these are plastids slash chloroplasts. Also, we can't forget the Golgi apparatus, or simply the Golgi body. And then we need the mitochondria. Multiple mitochondria we can have. So now this is the mitochondria. So now we have the simple diagram of the plant cell. Oh yeah, I forgot one thing. Vacuole, that's the main thing. So now we have the diagram of the plant cell and animal cell done, but we're forgetting one thing. If we move back, we forgot the cytoplasm. Normally everyone just forgets to put the cytoplasm inside their cell diagrams, but that is actually the most important thing. It's this cytoplasm which is actually keeping all these cell organelles together. Without this cytoplasm, I might have the nucleus here, my mitochondria might be all the way over here, my Golgi body might be all the way over here. So it's this cytoplasm which pulls everything together. The cytoplasm is needed to keep everything intact inside this cell wall so that it's rigid, so that the nucleus can't move, the mitochondria can't move, so that they're unable to move basically. So this is why we need the cytoplasm. So I'll just mark it real quick. This is the cytoplasm. Also, we'll mark it on our animal cell as well. Cytoplasm. This is really a common mistake everyone makes. They just forget the cytoplasm. So, but it's required to put the cytoplasm inside your animal and plant cell diagrams. And also one common mistake is that people think that these ribosomes over here, these ribosomes are only present on the rough endoplasmic reticulum. But actually, there are ribosomes throughout the cytoplasm. These all are ribosomes throughout the cytoplasm. So even in the plant cell, we will have the ribosomes throughout the cytoplasm. So these are the two basically common mistakes. We just forget to label the cytoplasm and we forget to make these dots in the cytoplasm which indicate ribosomes. Now we have the diagrams out of the way. We'll compare and contrast the animal and plant cell. So now this is the most common difference. The animal cell does not have a cell wall. Plant cell has a cell wall. In the animal cell, the nucleus is in the center. If you remember, we drew the nucleus in the center. But in the plant cell, because the vacuole is so big, the nucleus is actually pushed offside. It's pushed off to the side in the plant cell, the nucleus, because the vacuole takes so much space. In an animal cell, we have multiple vacuoles. In the animal cell, we have multiple vacuoles. In the plant cell, we have a single central large vacuole. Plastids are absent. We don't have chloroplasts in animal cells. This is obvious because animals can't make food. Plant cells, however, have plastids. They have chloroplasts. They undergo photosynthesis. They make their own food. And lysosomes are always present in animal cells because we don't have that tough outer cell wall, so we can't restrict everything from getting into the cell. However, plant cells have the tough outer cell wall, so that's why they prevent most of the things coming inside the cell. However, if by chance something goes inside the cell, they end up inside the vacuole, and they're just designated spots inside the plant vacuole in which these wastes accumulate. So this is why lysosomes are not generally present inside plant cells, but you might see them here and there. And the last topic of this entire chapter is cell division. So there are two types of cell division. There are two ways in which cells can divide. The first one is mitosis, and the second one we'll look in the next slide. So mitosis is a type of cell division in which one mother cell divides into two identical daughter cells. Basically, one mother cell will divide into two daughter cells, and they are identical. They're exactly the same. The daughter cells have the same number of chromosomes as the mother cell. So if the mother cells has, let's say, X chromosomes, then the daughter cells will also have X, and this one will also have X number of chromosomes. Now this type of division as X stays X, X stays X, is known as equational division. There's no loss in chromosomes on dividing. The number of chromosomes stays same even after division and before division. This type of cell division is used majority by the cells for growth and repair. So mitosis, you can remember, is used for the growth and repair of cells. So now I'll quickly explain how mitosis happens. So over here we have the mother cell. It has DNA and everything inside of here. So the first step, so this is beginning. This is the beginning. So the first step, step 1, is to duplicate or replicate the DNA inside the mother cell. So what will happen is the nucleus will divide into two parts. First, the nucleus itself will divide into two parts. So first the DNA will duplicate inside this original mother cell. Then the nucleus will form, they will form two nucleuses. This will be the nucleus of daughter cell 1. This will be the nucleus of daughter cell 2. And then after the nucleuses get separated, so we will have this situation inside the cell, two separate nucleuses. Then the cytoplasm will divide. Then the cytoplasm will also divide in two. And then as a result, we will get one daughter cell and we will get another daughter cell, and they have same number of chromosomes. So this is the main thing about mitosis, is that they have the same number of chromosomes. How can they get the same number of chromosomes? As they first duplicate everything, then they divide the nucleus, then they divide the cytoplasm. So this is the main process of mitosis, the first type of cell division. The second type of cell division is meiosis. Meiosis, whatever you want to call it, it's really tricky. So in this type of cell division, there's one mother cell as before, but in reality in this, what happens is you will get 4 daughter cells. So two divisions happen in this. This is step 1. This was just mitosis. However, in meiosis, you do the division again. So then you will get 4 daughter cells. So you will have one mother cell. This will divide into 4 daughter cells. So this is the main thing about meiosis. It divides into 4 identical daughter cells. The main point in meiosis is that the daughter cells have half the number of chromosomes as the mother cell. I'll explain how this happens in the next slide. So then, as this has half the number of chromosomes, meaning if we have this as the, oh sorry, I don't know what's happening. Yeah, so we have this as the mother cell. Excuse me, sorry for the inconvenience. Yeah, we have this as the mother cell. It has X. So what's going to happen is the first division, it will have X, but then upon the second division, it will be X by 2, X by 2, X by 2, X by 2. So this is what happens in meiosis. The first division, it's X and X, but then after that it reduces to half the amount. The number of chromosomes is half as the mother cell. So these were the 4 sorry, these are the 4 daughter cells in the end. Now this is known as reductional division. As the number of chromosomes get half, they reduce, and this type of cell division is used by the reproductive cells or gametes. I'll explain this in the next slide. So how does meiosis happen? The first step is exactly the same as mitosis. So meaning the nucleus first divides into 2. This nucleus gets divided into 2, and then the cytoplasm gets divided. This is the cytoplasm, and then we get 2 cells. Now initially there is equational division. These both daughter cells in the first step have X number of chromosomes. This also has X. The mother cell had X. But after this, when this divides, it does not duplicate the nucleus as in this. Meaning, it divides the nucleus just in half. Initially what we did, we duplicated the number of chromosomes. First, in mitosis, we duplicated the number of chromosomes, and this is what happens in the first step. However, in the second step, what happens? We don't duplicate the number of chromosomes. The number of chromosomes stay the same, but the cell is dividing in 2. So what happens? The number of chromosomes will reduce in half. As the number of chromosomes we didn't duplicate, but the cell got divided in 2, so the number of chromosomes get divided in half. So this is also what happens for this cell. Now you might be asking, why do we need half the number of chromosomes for reproductive cells? Well, as you remember, for reproduction to happen, the male gives a cell and the female also gives the cell. Both of them give a cell, and that forms a baby. However, if the male gave X, female gave also X, then the baby will have 2X. So that doesn't make sense. So this is why the male gives half, the female gives half, and then the baby gets the total X. So this is why in meiosis, which happens in reproductive cells, we get half the number of chromosomes. We essentially require half the number of chromosomes in meiosis. So now a quick recap. We drew the animal cell, we drew the plant cell, we discussed the differences and how the animal cell doesn't have a cell wall. In the plant cell, the nucleus is shifted to the side, etc. And then this was the new thing today. We discussed the 2 types of cell division: mitosis in normal cells and meiosis in reproductive cells. We discussed equational division and reductional division. So these are the 2 things we discussed today, and that will sum up this chapter, and that will be the end. So in the next lecture, we'll start off with tissues and what are tissues and the type of tissues in plants and animals. So that's it for today, and I'll see you next time.